Sir, good morning, sir.
Good morning.
Sir, good morning, sir. Sir, joined, sir. Varaprasad garu joined, sir.
Yeah. Sir, good morning.
Good morning, Andy. Good morning.
Good morning, sir.
Our Honorable Vice Chancellor, sir.
Yes, good to see you, sir.
Sir, how are you?
I'm doing fine.
Yeah, మిథిలా గారు మా క్లాస్మేట్ బాగా చెప్తా ఉంటారు మీ గురించి. సో యాక్టివ్, ప్రోయాక్టివ్, బాగా మంచిగా అన్ని చేస్తూ ఉంటారు అని చెప్పేసి. అండ్ దట్ ఈస్ ట్రూ. ఐ హావ్ సీన్ యువర్ బయోడేటా. ఓ సో గుడ్, ఎక్సలెంట్ సార్. కంగ్రాచులేషన్స్. హ్యావ్ ఎ నైస్ బయోడేటా. దట్ షోస్ యువర్ హార్డ్ వర్క్ అండ్ ఎవ్రీథింగ్. ఎస్.
Thank you, sir. Thank you.
Sir, can I start, sir? Can I start, sir?
Sir, good morning. Honorable Vice Chancellor garu, good morning.
Sir, good morning, sir.
Good morning, Dr. Dhananjaya Rao garu.
Good morning. Good morning.
Also, good morning and welcome to Dr. Varaprasad garu.
Namaste, Andy. Namaste. Good morning.
Good morning. I am Dr. Srinivasu, Registrar.
Nice to meet you, sir.
I think I have seen you at Rajendra Nagar
Andy.
during 91 to 93.
That's correct.
I was doing my PhD there at Rajendra Nagar.
That's neat.
Nice to have with you.
Thank you, sir. Thank you.
Right.
Sir, shall I start, sir? Wait for...
ఓకేనప్పా, స్టార్ట్ చేయండి.
Okay.
అండ్ అగైన్ ఐ రిక్వెస్ట్ రిజిస్టర్ గారు,
Sir.
వాళ్ళందరినీ కూడా వీడియో ఆన్ చేసుకోమని చెప్పండి. ఇట్స్ నాట్ ఫేర్.
Yes, sir. One request to all the participants to kindly switch on to the visual mode. All the participants, there are about 25 participants are there. My request to all the participants is to kindly switch over your video so that our chief guest and our honorable vice chancellor will be able to see you all.
Authentication request.
Okay, sir. Sir, good morning, all of you. Welcome to Dr. YSR Horticulture University, Venkatramanagudam, West Godavari District, Andhra Pradesh, for the 23rd Global Connect series in collaboration with the Kansas State University, USA, on impact of heat stress on horticultural and food crops. Good morning, sir. Good morning. Welcome to this 23rd Global Connect series. 3 years back it was started to arrange the lectures by the international eminent scientists to update the knowledge to the students and faculty on the latest technologies and what happening in the international and India. And so welcome, sir. Welcome to our chairman, Dr. K. Gopal Garu, Honorable Vice Chancellor, Dr. YSR Horticulture University. With his dynamic leadership and vibrant dynamic leadership, we are conducting 23rd Global Connect series. Thank you very much, sir, for giving permission to conduct 23rd Global Connect series. And today, our guest speaker, Dr. P.V. Varaprasad, Pagadala Venkata Varaprasad, University Distinguished Professor, R.O. Cruise Endowed Professor and Director, Center for Crops, Climate and Communities, College of Agriculture, Kansas State University, USA. Welcome, sir. Welcome. And thank you for accepting for as a guest speaker, today's speaker, for giving the 23rd Global Connect series. And thank you very much, sir. Welcome to Dr. YSR Horticulture University and all our university officers, Dean of Horticulture, Dr. Naram Naidu Garu, PG Dean and Director of Research in Charge, Dr. K.T. Venkatramana Garu, Director of Extension, Dr. Govindarajan Garu, Registrar, Dr. B. Srinivasu Garu, Dean of Student Affairs, Dr. Surya Kumar Garu, Controller of Exams, Dr. Prasanna Kumar Garu, University Librarian, Dr. Ramanandam Garu, Director, Planning and Monitoring Cell, Dr. Vijay Padma Garu, Controller, Dr. Srinivasa Subramanian Garu, Estate Officer, Sri Ramana Garu, all Associate Deans, all Heads of Research Stations and KVKs, and all Principals of Polytechnic Colleges of Horticulture. Welcome today's guest lectures of 23rd Global Connect series. And before inviting our Honorable Vice Chancellor, just I will give the brief introduction about our today guest speaker, Dr. P.V. Pagadala Venkata Varaprasad Garu. And P.V. Varaprasad Garu obtained BSc Agriculture and MSc Agriculture from Andhra Pradesh Agriculture University, Hyderabad, and PhD from the University of Reading, United Kingdom. His research focus on understanding responses of crops to changing environments and developing best management strategies to improve and protect yields. He has active research, education, outreach activities around the world. He has published over 512 peer-reviewed publications, 455 journal articles, 57 book chapters, 5 edited books, which are cited over 36,000 times with an H index of 98 and I-10 index of 330. He is listed in the top 1% of highly cited and influential scholars around the world for the last 3 successive years. He received extramural funding of over $200 million for his activities. He graduated 25 students, 15 PhD and 10 MS as major advisor, 70 students, 30 PhD and 40 MS as a committee member, and trained more than 220 visiting scholars from over 15 countries. For his research, education, and leadership, he received over 60 competitive awards and recognition from multiple organizations around the world. He is a fellow of American Society of Agronomy, Crop Science Society of America, and American Association for Advancement of Science. He is a former president of Crop Science Society of America. He was recently elected to serve as the president-elect 2025 of the Council of Agriculture, Science and Technology. And Sir is also adjunct professor. He worked in various capacities, presently director of Center for Crops and Climate Communities. And Sir is also adjunct professor in Tamil Nadu Agriculture University, SRM Institute of Science and Technology, Tamil Nadu, and University of Philippines, Sher-e Kashmir University, Agriculture Science and Technology, Jammu and Kashmir, India, and adjunct professor, Royal University of Agriculture, Cambodia, and Sri Konda Lakshman, Telangana State Horticulture University, Hyderabad. And Sir is having many fellowships, elected fellow of 2014 American Society of Agronomy, 2015 Crop Science Society of America, 2016 American Association for the Advancement of Science, and 2023 Indian Society of Plant Physiology, 2024 National Academy of Agriculture Sciences. And Sir having many awards, received many scholarships and awards also, and Visionary Scientist for African Agriculture 2024, ASA, CSSA, North America, USA, and Certificate of Appreciation 2024, Royal University of Agriculture, Cambodia, Lifetime Achievement Award 2024 from Bihar Agriculture University and Excellent Vision Foundation, India, and KK Nanda Memorial Award 2023, Indian Society of Plant Physiology, India, Lifetime Achievement Award 2023, NutriHub, India Institute of Millet Research, India, and Distinguished Service Award 2023, Crop Science Society of America, and International Crop Science Award 2023, Crop Science Society of America, and Global Scientist 2023, Agriculture and Environment Technology of Development Society, India, a working group member 2223 from National Climate Change Roadmap Working Group, and Outstanding Unit Award 2022, International Program, Kansas State University, Ahuja Agriculture Systems Modeling Award 2022, Soil Science Society in America, and President 2021-22 Awards Crop Science Society America, University of Distinguished Professors, Kansas University, President 21-22, Sigma 11 Chapter KSU, International Agronomy Award 2020, American Society of Agronomy, and Commissioner 2021, Commissioner on Sustainable Agriculture Intensifications. Not only this, to read it takes a lot of time. Sir,
That's enough, sir. Thank you.
internationally
Thank you.
recognized excellent scientists, sir. Congratulations from Dr. YSR Horticulture University. And we are very fortunate to have as a guest speaker today. Now I invite our Honorable Vice Chancellor to speak on this occasion, Dr. K. Gopal Garu. Please, sir, welcome.
Sir, welcome to this Global Connect series. And it was started years back. And yes, on important topics, we invite guest speakers all over the country and also from outside. So here, you know, our university, it is young, 15 years, almost 16 years old. And we have 5 colleges, 4 KVKs, 22 research stations, and 11 polytechnics. We have good infrastructure and good research facilities in most of the research stations and major colleges where we have PG and PhD. We have around 60% of the faculty positions filled, so we have a good faculty strength. So here, what we need and expecting from Professor Sir is we want to have a very strong MOU with you and with your university, especially for student research projects and student exchange, and again faculty exchange programs, and writing collaborative projects. That we want. And again, second one, we want to have wherever it is possible, we have to talk in depth about it. That is regarding having twin degrees programs or sandwich programs, degree programs. So we want these things. We want to start mostly from next academic year, this degree programs. And the student exchange and all, we can, we have signed MOU and already we have sent to you for further action there at your end. So we want to have a very strong collaboration with you and your university and with your colleagues. And we are happy that one of known international scientists from our country is there, so that bonding will continue, and we want to have a very strong bonding. So with this, I request Director International Programs to continue. All the best, sir. So I once again request very good and meaningful MOU and MOAs between our two institutions for the students, staff, and mutual other research projects. Yes, sir. Thank you.
Thank you. Thank you.
Thank you, sir. Thank you for your valuable speech. And already we have signed MOU, sir, and that both institutes have signed already. And sir is coming in June to our university, what he told just yesterday. And anyhow, sir is coming on June, and we'll arrange one special program also, and function and all, one talk also from the sir live. Definitely, sir. And now I invite our guest speaker, Dr. P.V. Varaprasad Garu, to speak on this occasion. He is a university distinguished professor, who is endowed professor and director, Center for Crops, Climate and Communities, College of Agriculture, Kansas State University, USA. Sir, please.
Yes, thank you very much. Good morning, everyone. Thank you very much for your kind invitation, and also for your kind and elaborate introduction. Appreciate that. And also special thanks to Honorable Vice Chancellor Gopal Garu for giving me this opportunity to speak to you, and also the Director of the International Programs, the Dr. Dhananjay Garu. And also I would like to invite or thank all the university officers and also administrators, faculty members, students, and scholars who have joined today to listen to me. And as I said, the topic of my presentation is going to be the impact of heat stress on horticultural and food crops. And I'll take about an hour, and then we'll have significant amount of time to make sure that we have enough time for questions.
Sir, kindly increase that volume.
Yeah,
Bit-
okay.
it's
Can you
bit
hear me
low.
now?
Yeah,
better?
yes, now it's good. Yes.
All right, sounds good. So before I go into my talk, I would like to give a brief introduction of Kansas and Kansas State University. Kansas is right in the middle of US, right in the middle. It is geographical center of the US. And within Kansas, Manhattan is on the northeast side of the state. And Kansas has a population of about 2.9 million people. Economy is primarily driven by agriculture. And our cropland is about 28.2 million acres. Only 10% or less of that is irrigated, about 2.76 million acres. Kansas is the first land-grant university established in the US in 1863, and it's the #1 producer of the wheat and sorghum in the country. About 25% of the US wheat comes from Kansas, and almost about 60% of the sorghum also comes from Kansas. And our College of Agriculture is ranked always in the top 5 in the country. And more importantly, Kansas State University has a long history of working in India, and specifically in Andhra Pradesh. Kansas State University led the efforts to establish the land-grant system in India. About 30 to 40 faculty members from Kansas State University came in and out of India for 16 years, starting from 1956 to 1972, to create an education system that continues till now, which is called as a land-grant system, where the university conducts research, education, outreach, and extension activities. And it established the Andhra Pradesh Agriculture University in 1963. And at that time, when the report was finally concluded after the inauguration of the Andhra Pradesh Agriculture University by then Honorable President of India, Sri Lal Bahadur Shastri Ji, and the report concluded that the procedures used to establish Andhra Pradesh Agriculture University. Can you please mute?
Please, one request, other participant, please, don't cross-talk when the program is going on. Please mute your these systems.
Great.
Well,
Thank you so
yes,
much for
please.
that. Appreciate it. Please, if you can kindly mute your computers or laptops or phones, that would be very good. So that report, when it concluded, it was saying that time will give the true answer. If India continues to move ahead as a democratic nation against the mighty odds she faces, we can then conclude that our work has been well done. So that was the note in 1963. Year 2025, 60 years later, all the components of agricultural land-grant agriculture system still operational in over 70 agriculture universities in India, and it is food insecure, the 5th largest economy in the world, and the leading democracy in the world. So that's really a true testament of the success. And early this year in March, it was a privilege to sign an MOU between the YSR Horticulture University and Kansas State University. This was signed on 3rd March of this year by your Honorable Vice Chancellor, Dr. Kuruba Gopal Garu, and our Honorable President, Richard Linton of Kansas State University. And this MOU is for cooperation in research, education, and cultural activities. And also there will be opportunities to exchange of scholars, students, faculty, and knowledge sharing. And I truly want to mention and reciprocate what you have said, Honorable Vice Chancellor Gopal Garu, that we want to make sure that this memorandum is not just a signature, but actually we will be able to do some productive work in terms of student exchange, faculty exchange, and also from some collaborative research projects. Now I'll dig into the more scientific talk of today to talk more about the impact of heat stress on horticulture crops. And this will be the outline of my presentation. First, I would give you a brief overview of the greenhouse gas emissions and the temperatures. We'll look into the past trends and also how the future projections look like. And then we'll talk about the impact of heat stress on reproductive processes and yield, and what are the most sensitive stages in some of the horticultural crops. And I would take the case study of tomatoes, peppers, and coconut, and also give you some impacts of short episodes of heat stress and sensitive stages for the key grain crops, which will include rice, wheat, sorghum, and pearl millet. And then we'll talk a little bit about the mechanisms of reproductive failure or seed root set under heat stress, and then impacts of season-long effects of heat stress, and what are the temperature thresholds for different key grain crops, and then what is the current genetic variability, and what are the opportunities for stress tolerance, and then how can we manage the heat stress. I'll provide an overview, one slide, giving you some agronomic practices which can be used to manage the heat stress, and what are the key breeding approaches to develop heat-tolerant varieties of these crops. And then we'll have the conclusion and what is the future research direction. So now first, let's look into the current greenhouse gases, emissions, and temperatures. What were the past trends and the future projections? So this visual gives you the greenhouse gas emissions trend for the last many decades. We have 4 major greenhouse gases: carbon dioxide, methane, nitrous oxide, and sulfur hexafluoride. The concentration of these all 4 greenhouse gases have been significantly increasing over many decades. At the end of December 2024, our carbon dioxide concentrations were about 426 ppm. Methane was 1938 parts per billion. Nitrous oxide was 338 parts per billion, and sulfur hexafluorides were 11.99 parts per trillion. And as you know, all of these are called greenhouse gases because they can trap the heat. And because of that, the global surface temperatures of Earth has significantly increased. This visual on the left shows you the temperature change in the last 50 years when compared to the average of 1951 to 1978 as a baseline. As you can see, pretty much most of the Earth has significantly increased the temperatures. That temperature increase has been between 1 to 2 degrees Celsius in most of the areas. And the visual on the right gives you the global average temperatures from 1850 to 2023. Year 2024 was the warmest. That was the first time we actually reached the temperatures of 1.5 degrees Celsius warmer than the beginning of last century. Those of you who are following the COP meetings, our goal is to keep the temperature warming below 1.5 degrees Celsius by end of this century. And if not, at least we need to keep it under 2 degrees Celsius. And now if you look at the global temperature anomaly, this is the global anomaly from almost beginning of 1900s to now. The Earth's surface temperature since 1980 was significantly warmer compared to that and average until that point of time. And in recent years, we have seen much more temperature anomaly, especially in the last 4 to 5 years. The temperature anomaly has been very high, close to almost 2 degrees Fahrenheit. And if you look at the heat waves in the United States, when I say heat waves, we usually measure the heat waves in frequency, distribution or duration, and season and intensity. Frequency meaning how frequently we are getting these heat waves, and what is the duration of that heat waves, and then what is the season like, is it hot, and then what is the intensity, how much intense heat waves are observing. So if you can look at it, each bar is from decade, from 1960s to almost 2020s. All of these indicators, frequency, duration, season, and intensity, have significantly increased over the decades, and much more in the last 2 decades or the last decade compared to that previous years. And this visual shows you the hot days and night temperatures in the United States in the land area. And this is data from 1910 to almost 2020. As you can see, there has been significant increase in both daytime temperature, daytime maximum temperature, and the nighttime minimum temperature. However, the percentage of land area is greater on the high nighttime temperature. So if you look into the future, we are likely to get more hotter nights compared to that of daytimes. And that will have a significant negative impacts on productivity of our horticulture and also grain crops. And this visual is the temperature trends or the heat waves in India. This is the data from beginning of 19th century to almost 2015 or 2019, actually. As you can see here, this is an average mean annual temperature of the country. And there has been significant increase from 1980 onwards, as we saw for United States as well. And then specifically in June of 2019 was almost 26.5 degrees Celsius. And then if you look at the number of heat waves, which is designed as more than 40 or 45 degrees Celsius, the number of degree heat wave days in India across all the states is been significantly increasing in the last 3 to 4 years. And specifically in 2024, we saw about 554 heat wave days in India. That was probably the hottest when compared to that of 2010. So these number of heat waves have been rapidly increasing over the past years. And how does the future look like? In the future, we expect that both not only the mean temperature is going to increase, but also that there will be much more likelihood of getting extreme weather events, specifically hot or extreme hot weathers. And then that will also lead to further increases in the temperature. So it is expected that if we continue business as usual, the temperature increase by end of this century could be anywhere between 2.8 to 4.5 degrees Celsius warmer than the beginning of this century. And that's for the temperature. And if you look at the precipitation or the rainfall, it is expected that will be variable. In some regions of the world, we'll see more rainfall, but the higher evapotranspiration due to hot temperature will lead to significant water stress or drought stress. And of course, the different models predict different projections in different regions. So overall, there'll be increase in the mean temperatures, but also increase in the rainfall, but the drought periods will be, or the water stress periods will be more because of the higher demand for evapotranspiration and evaporation due to hot air temperatures. And now we'll go into these 3 specific crops and look at what the heat stress can do to these crops. We'll start with tomato, then we'll go to pepper, then we'll go to coconut, and then I'll give you a summary of some of the grain crops before moving into some of the opportunities for agronomic and genetic improvement of these crops. So let's start with tomato. We'll talk about the impact of heat stress on fruit set and what are the most sensitive stages and what are the thresholds for temperature response for tomato. Before I go into the detail, I would just like to bring everyone up to the knowledge for the crop growth and development. As we know that most of the crops have 3 phases: vegetative phase, reproductive phase, and the maturation phase. And these are the growth stages of tomatoes. Starts with germination, emergence, leaf establishment and leaf development, stem elongation and shoot development. Then we go to the floral bud development, flowering, fruit set, and then fruit development and ripening, and then senescence and the death of the plant. Not all the stages of crop growth and development are equally sensitive to temperature stress. Some of these stages are more sensitive when compared to others. And if we know which is the most sensitive stage and what are the processes it impacts, we have an opportunity to manage our crop by adjusting our planting dates, selecting the crop which is of right duration so that we can avoid occurrence of temperature stress during the most sensitive stages. In general, the reproductive stages of crop development, that would be from initiation of the floral bud development through flowering, through the fruit set, are generally more sensitive to heat stress compared to that of later stages of fruit development. And I'll give you more specific details on that. So if you look at the reproductive stage, as I said, reproductive stage of development is much more sensitive, which is still a longer phase. So if you look at more carefully about the timeline of tomato in terms of reproductive development stage, if you look at the transition from the floral bud development to flower opening to anthesis to a few days after anthesis to maturation of the fruit, it can take anywhere from 0 to 1 to 2 days for the floral bud development. Flower opening could be there for 3 or 4 days, and then anthesis happens. And then after anthesis, the seed is set, and the fruit is formed, and the fruit matures. So these are the different stages of within that reproductive stage in terms of developmental stages for flowering. And if you go a little bit more detail into that, what is happening at the floral bud development, there are two specific processes which are happening in the floral bud development, which is the gametogenesis, which could be the development of the pollen, which is the male gametophyte, and also the development of the ovules, which is the female gametophyte. So in this case, we are looking into the microsporogenesis of the pollen development. So that during the microsporogenesis, the floral buds go through the meiosis stage and then reach the tetrad stage, that's microsporogenesis. And after that, the microspores are released and it goes through the microgametogenesis, where this pollen development occurs, mitosis, and then it mature pollen. So these are the things which are happening. If we're able to identify which is the most sensitive stages of these periods, then we can develop management strategies to avoid the heat stress. So now let's look into the impact of heat stress. So if you look at the controls in this experiment, there was control is 28 by 22 degrees Celsius. 28 is the daytime maximum and 22 is the nighttime minimum temperature. And this is the high temperature stress, which was 32 degrees Celsius by 26 degrees Celsius. If you look at the control flowers, they're much more nicer. And if those flowers which are exposed to heat stress, you often see the exertion of the stigma. That means stigma is exerted. That means stigma is at a higher level compared to that stigma is at a higher level compared to that of the male flowers. Therefore, the pollination does not occur well. That means the pollen reception by the stigma is lower, which impacts negatively the pollination of that crop. And this shows you that in general, when you have higher temperature stress, the stigma exertion occurs. And then also the number of pollen grains which are germinated are very low. Not only the pollen development, total number of pollen grains is decreased when you have stress occurring during the floral bud development. The germinated pollen grains are lower. The viability of the pollen grains is also lower. And the number of non-viable pollens are higher. So when you lose the pollen viability and the pollen is sterile, the fertilization is not going to happen. So that is one of the mechanisms. And how does the pollen viability is lost? What's the mechanism which is impacting the pollen viability? This visual shows you the top row is for the control plants, which is 26 by 19 degrees Celsius. And the bottom row is for the heat stress plants, which is 36 by 26 degrees Celsius. As you can see in the high temperature stress, what it does is it interrupts the pollen development. It causes early degeneration of the tapetum tissue. Tapetum tissue is the one which provides nutrition to the developing microspore. So if the developing pollen grains does not get the nutrition, then they are non-viable or sterile. That leads to loss of fertilization. And because of that lack of degeneration, early degeneration of the tapetum tissue. And the second mechanism is associated with the starch. If the temperature stress, under high temperature stress, the starch metabolism is impacted. And what happens is the pollen grains has the lower sugar levels. And if you have the lower sugar levels, then the pollen is not able to survive. And then this leads to pollen sterility as well. That eventually will lead to decreased fruit set. The heat stress does not generally affect the total flower number. It decreases the pollen numbers and also decreases the pollen shed and the pollen viability. And when that happens, it results in lower seed set. And if you look at the threshold temperatures, typically with increasing, these are the growth temperatures with the mean daily and daily temperature, which is an average of Tmax and Tmin. With increasing temperature from 14 degrees Celsius to 26 degrees Celsius, the number of fruit set is decreased. And then number of flowers are decreased, but not too much. But the development rate or the development time, total development time is decreased because of the higher accumulation of the growing degree days at a higher temperature. So all of this leads to the decreased fruit size. And that's a summary of the story about tomatoes. Now we'll talk a little bit about peppers. So when I'm talking about peppers, I'm talking about two types of peppers here. One is the bell pepper, and another one is the chili pepper. And I'll give you a few examples of both of these species. And just like the tomatoes, the peppers also have a vegetative stage, first germination, emergence, vegetative stage, flowering, fruit set, and then food growth and maturity. And the story is almost similar to that of the tomatoes. Generally, the reproductive stages of crop growth and development are much more sensitive to heat stress. And then specifically, the 2 time stages is most sensitive. So in this case, the high temperature stress was 33 degrees Celsius. And these 4 plots shows you the impact of high temperature stress on the percent fruit set at meiosis, at tetrad formation, at young microspores, and at mature microspores. If you look at this data, of these 4 stages, stage 1, which is the meiotic stages, is the most sensitive. That's where you see the maximum reduction in the fruit set. The tetrad stage is not that impacted, and this young microspore stage is also not that impacted, but the mature microspore stage is significantly impacted, where it decreases the seed set. So there are 2 specific most sensitive stages within the floral bud development, which happens between 2 to 5 days before anthesis. One impacts the meiosis stage of the microspores, and then the mature microspore stage. This impacts the viability of those microspores, which is in this case the pollen. And here you can see that at normal temperatures, the pollen grains is nicely intact, and then they are viable. Whereas at a high temperature stress, the pollen grains are abnormal, and then also the meiosis is not happening that well, and the pollen grain is not fully developed. So that's how the heat stress impacts the viability or the pollen development stage. And the loss of pollen viability, for tomatoes, I mentioned that it is about the starch metabolism or sugar metabolism, but specifically it is that activities of the sugar transporters, the hexokinase and the fructokinase activity is the one which is downregulated under high temperature stress, both in the pollen grains and the anthers, meaning that the fructose and the glucose content is decreased, and that is because of the activity of the hexokinases and the fructokinases, which are the ones which are responsible for these activities. And we've also identified that the hexose transporters, the transporters which actually move the starch or the sugars from anther walls into the pollen grains, are downregulated, and that leads to the loss of pollen viability. And in terms of the most sensitive stage, and these are different stages of development which were exposed to heat stress. On the left is a visual diagram of the chili peppers itself. In this case, the Shishota peppers. And the right side gives you the data on the fruit set, percent seed set on the fruit weight itself. In this case, the control was 30 by 22 degrees Celsius. And then they had the temperature stress exposed to different stages, from anthesis to 5 days after anthesis, anthesis to 10 days after anthesis, and then 10 days after anthesis to 30 days after anthesis. And the last one was 30 days after anthesis to and harvest. And if you can look at it, the most impact was happening, the maximum reduction in the seed set from 36 degrees Celsius to 12.4 and 13.4 was happening when the stress was imposed at anthesis. to 10 days after anthesis. Whereas if the stress was imposed at a later stages, 30 days after anthesis to harvest, there was not much significant impact on the seed set. That tells us that the pre-anthesis stage and the anthesis stage is more sensitive to heat stress. And that was the story on the peppers. Now let's quickly move on to the coconut, which is a more of a perennial crop. And this work was done by Dr. Hebar. He is currently the director of the Indian Council of Agriculture Research, the CPCRI, Central Plantation Crop Research Institute at Kasaragod. And he visited my lab as a Borlaug scholar 10-15 years ago. And then I was doing this type of work on the grain crops. So he was very much interested to continue that work and then do it on coconut. So he looked at the temperature response of or impact of high temperature stress on coconut. And he screened multiple varieties or genotypes of coconut from different origins, some from India, some from Africa, Malaysia, Philippines, Vietnam, and looked at how much pollen germination they had. And there was significant genetic variability in terms of pollen germination at different temperatures. So he collected the pollen from these plants and then exposed them to different temperatures in incubators and measured the pollen viability. As you can see, one of the tolerant variety was named as WCT, and even at very high temperatures, there was some good germination. As you can see, at 35 degrees Celsius, there was still some good germination here. And the susceptible variety was MYD, and they did not see much of a germination at high temperatures there. So there is genetic variability for high temperature stress in terms of pollen germination in coconut. But of course, this was done in vitro. So he wanted to do in vivo in the plant itself. So he was very smart and excellent scientist. So what he did was he developed a technique where actually he put a canopy on the male and the female flowers on the coconut plant live, and then pollinated them. And then he actually controlled the temperature and the humidity within this small chamber. And then he pollinated, and then he looked at the in vivo or in-plant pollen germination and the pollen tube growth. It's a very difficult technique, but he really professionally did it. And he was able to document the differences among the various coconut genotypes and also quantified the rate of pollen tube growth in vivo in the plant. So you can see on the visuals on the side, on the right side, after the female flowers was pollinated, and then he actually measured the pollen tube growth, how fast it was growing and how long it was growing. So if you look at the data, he had the ambient control and the chamber control. Ambient was about 34 degrees Celsius, and the chamber control was 35 degrees Celsius. And by those chambers, he was able to increase the temperature within the chambers by about 3 to 4 degrees Celsius. And then he maintained the relative humidity in those chambers, one at 55% relative humidity with 39 degrees Celsius. And then another one was 39 degrees Celsius, but 65% relative humidity. And then he measured the pollen tube growth in vivo in the plant on day one, day 2, day 3, and day 4. As you can see, the ambient control, the pollen tube growth was pretty good. And for example, in this case, it was 19. And then the control chamber was 12.89. But when the temperature was high, 39 degrees Celsius, with a relative humidity of 55%, there was no germination of the pollen tube growth. And when the relative humidity was a little higher, 65%, there was pollen germination, but the pollen tube growth was not that great. So that clearly shows that both the temperature and relative humidity can have a significant negative impact, not only on the pollen germination, but also pollen tube growth. So then what he did was he actually collected the male and the female flowers, and then this was in vitro, meaning that he actually created this, that was on the plants. And here he wanted to get a temperature response of these fruit sets. So what he did was he developed this small chambers which allow, and he was able to control the temperature and the relative humidity in these small chambers. And he actually collected the female flowers and then actually pollinated them and then exposed them to different temperatures in different chambers and looked at the pollen tube growth in the on the female flowers. And in this case, as you can see, under both relative humidities and the cooler temperatures of 29 and 29 degrees Celsius, there was not much negative impact on the pollen tube growth. This data is in millimeters, and so was the previous one as well. But with increasing temperatures, especially at the lower relative humidity, the pollen tube growth significantly decreased. But when the higher relative humidity was there, the temperature above 33 degrees Celsius only decreased the pollen tube growth. Whereas at a lower relative humidity, that the pollen tube growth decreased soon after 29 degrees Celsius. Even at 31 degrees Celsius, they saw lower pollen tube growth. That's a summary of the three major horticulture crops. Hopefully, I gave you enough information that reproductive stages of crop growth and development for all the three horticulture crops was much more sensitive to heat stress. And specifically the two stages, the floral bud development when the gametogenesis happens was the most sensitive stage, which impacts the pollen viability. And then at the time of flowering, it impacts the pollen tube growth and pollination. Now I will transition you to the major grain crops, and I'll give you examples of 4 crops. Wheat, actually I have five crops. I'll give you data from wheat, sorghum, rice, and also peanut or groundnuts. So first we'll look into the short episodes of heat stress. As I showed you in the climate data, there'll be much more extreme temperature stress of shorter duration with higher frequency and high intensity. So it could be from anywhere from 1 to 2 days to 4 to 5 days, or maybe even few hours within a day. And then we'll talk about, some people ask that if 1 or 2 days of heat stress, is it going to have any negative impacts on the viability of the gametes or on pollination and fruit set? The answer is yes. And I'm going to give you data from multiple crop species. Bear with me for this particular visual. I have thought that I'll summarize it all in one nice visual to give you that information. So if you look at this visual, I have given you the data on the sorghum on the top left, and then wheat on the right, peanut or groundnut on the left bottom, and the rice on the right bottom. So if you look at sorghum, in this case, the optimum temperature was 30-20 and high temperature was 36-26. And I also give you a different duration of heat stress. For sorghum, 5 days, for wheat, 2 days, for peanut, 1 day, and for rice, 6 hours. So if you look at this one particular visual there, and you pick the top left on the sorghum, x-axis is the anthesis, the duration timing from anthesis, meaning 0 is anthesis, and on your left is the minus is the 5 days before anthesis, 10 days before anthesis, 15 days before anthesis, and on the right side is 5, 10, 15, 20, and 25 days after anthesis. And your y-axis is the floret fertility expressed as a percentage of control. So the control plant has assumed to be 100%, and then how much decrease from that control did the high temperature stress had. So if you look at this data, the plants were exposed to heat stress at these different stages for a duration of 5 days, and the data on floret fertility is shown. So as you can see, the maximum reduction in the floret fertility has happened at the time of anthesis and 5 to 10 days before anthesis. These 3 data points right here for the sorghum. Whereas anytime before that or after that did not have any significant impact on the floret fertility. That tells us that the 2 stages which are extremely sensitive are at the time of anthesis and then few days before anthesis. Now I wanted to pinpoint a little bit more. I have the data for all of these crops individually, but I'm just showing for the diversity, I'm showing you different data points for these crops. So for the wheat, it was 2 days of heat stress. That again refines it, that 0 is anthesis, And then about 8 days before anthesis is microsporogenesis or the gametogenesis happening. So it clearly identifies two stages. One is occurring at the time of flowering, and the second one is happening few days before flowering. And then this is the data for 1 day of heat stress on peanut, and that even further shows you much more detail. It clearly identifies 2 days, 0 days, 0 timing at anthesis, and the next one is about 4 to 5 days before anthesis. These are the two most sensitive stages for these crops. So what is happening during this? So any floret, any flowers which are exposed to heat stress, either few days before anthesis, about 4 to 5 days before anthesis for peanuts or groundnut, and at the time of anthesis, it negatively impacts the floret fertility. So what is happening during that stage? And I showed you the data for the horticulture crops. So what is happening few days before anthesis is the gametogenesis, meaning that the meiosis and the mitosis and the development of the pollen. So if you have heat stress happening during that time, the viability of the pollen grains is impacted. That means the pollen is not viable. Therefore, even if it was pollinated, it will not germinate, and it will not fertilize, and it will not form seed set. Therefore, it will not be forming any seed numbers or fruit numbers in the case of horticulture crops. So those are the two most sensitive stages. And the bottom right-hand side, I'm giving you the data for rice. Even exposing rice flowers for 6 hours of heat stress. In this case, the heat stress was 38-24 degrees Celsius. Even at the 6 hours of the heat stress, it can have significant negative impacts, but specifically when you have the temperature stress at the time of flowering. And there was genetic variability, and N20T is one of the most heat-tolerant heat-tolerant rice crop. So this clearly shows you whether it is horticulture crops or whether it's grain crops. The two most sensitive stages are happening few days before anthesis, when the gametogenesis happens, and which impacts the viability of the gametes, and at the time of flowering, which impacts the pollination, pollen germination, and the seed set. And this is the data on the pearl millet. Again, it's very clear at the time of flowering, you have the maximum decrease in the pollen germination, and also 10 days before anthesis, that's when the viability of the pollen grains is being determined. And in this case, it's with 2 days of heat stress. So that's the data on that. So this is a summary of all the crops, and I've not included the horticulture crops. Most of the data is my own research for wheat, sorghum, pearl millet, peanuts, dry bean, it's my data, and others are from the literature. So any of the grain crops, the two critical stages of which are sensitive to temperature stress is anywhere from 3 to 12 days prior to anthesis. That's when the gametogenesis, both male and female flowers, male microsporogenesis, and the female is megasporogenesis, which leads to loss of gamete fertility. And at the time of flowering, which impacts the pollen shed, pollen reception on the stigma, pollen tube growth, and the fertilization. So that's the impact. And what are the mechanisms which impacts the reproductive failure? And I'm just showing you the summary here. I have detailed information on all of these processes. So I've showed you the information that high temperature stress negatively impacts the pollen and the ovule function, decreases the fertilization, decreases the seed number or the fruit numbers, and therefore the fruit yield or the seed yield. There are 5 different mechanisms through which this is impacted. Number 1 is that under high temperature stress, the reactive oxygen species in the gametes is increased, and then antioxidant capacity decreases. And that leads to membrane damage, and that leads to loss of pollen function or the ovule function. So that's number 1 mechanisms. Number 2 is that under heat stress, there is a change in the phospholipids, because all of these pollen grains or the membranes have phospholipids. And then the unsaturation levels changes, and specifically the oxylipins are upregulated, which damages the membrane and therefore leads to the loss of pollen or ovule function. That's the second mechanism. And the third mechanism, as I showed you in the data, the pollen sugars are lower, and then the hexose transporters from the anther wall into the which transport the sugars from the anther wall into the pollens are downregulated. That impacts the nutrition, and therefore the pollen or the ovule viability is lost. And the fourth one is anatomical, and I showed you the information on that. It increases the thickness of the exine wall. It also damages the pollen development, and specifically the tapetum layer degeneration happens faster under high temperature stress. This tapetum layers provides the nutrition to the developing pollen, and therefore, if that is damaged, then the pollen viability is decreased. And the second one, and the last fifth one is the pollen reception by the stigma. And as I said to you that under high temperature stress, the stigma exertion happens. That means the stigma is taller than the anther. Therefore, when the pollen is spreading, it does not fall on the stigma, and the reception is decreased. And then your pollen tube growth is also lower. I showed you that information. So that is the mechanisms through which the pollen gamete viability or the reproductive failure happens in either horticulture crops or in the grain crops. So now let's look at the summary of the season-long temperature stress. So the data I showed you so far was short episodes of heat stress, anywhere from 5 days to 2 days to 1 day to 6 hours. And then I also spent my entire early career, I developed these temperature response curves for major food grain crops. And this is a summary of my early career work, about 5 to 6 years of work. So I'm showing you the temperature response curves for all of these major crops. All of this is my own data. And I also have data on mung beans and canola, and also on cowpeas as well. So, and I'm showing you the harvest index so that it captures the entire, what is happening entire season. So as you can see, wheat is the most sensitive, optimal around 16 to 18 degrees Celsius, and the ceiling temperature is 31 degrees Celsius. At 31 degrees Celsius, there is no seed set, there is no yield, therefore the harvest index is 0. And the blue one is for the rice, the red one is for the sorghum, the orange one is for the pearl millet, and then the purple one is for the dry bean, peanut is for brown, and then soybean is green. So as you can see, among the cereals, wheat is the most sensitive with a ceiling temperature of 31, rice followed 35 degrees Celsius, sorghum 38, and pearl millet 41 degrees Celsius. So among the millets, among the cereals, pearl millet is the most heat tolerant crop. And for the legumes, dry bean is the most sensitive, which is common bean 32, groundnut is 39, soybean is 40, and the mung bean and cowpeas were 42 degrees Celsius. So mung beans and cowpeas are the most heat tolerant in terms of legumes. So when you have the season-long temperature, how is that harvest index being decreased? So this is a summary of the overview of the how harvest index or the yield are decreased. So when you have high temperature stress, this is the whole plant response. So what happens under high temperature stress is that the canopy temperature goes up, and then you have higher vapor pressure deficits as well. And when your canopy temperature goes up, it decreases the photosynthesis and also increases the respiration. So the net carbohydrates which are available for growth is decreased because you are decreasing the photosynthesis and increasing the respiration. So the available carbohydrates is the gross photosynthesis minus the respiration. So this leads to lower carbohydrates available for the plant growth. And then on the higher temperature, just because of the accumulation growing degree days, the shorter is the growing season, meaning that from germination to maturity or emergence to maturity, the growing season will be decreased in the high temperatures. And if you're looking at the reproductive stages as well, the duration of the seed filling duration or the fruit growing period is decreased, which will lead to lower yields. And then I showed you the reproductive failure, which leads to lower grain number, and shorter grain filling duration or the fruit filling also decreases the fruit size. The combination of all of those leads to lower grain yield and the decreased harvest index. So that summarizes the impact of heat stress. So now I'm at 43-minute mark, so I'm going to wrap it up in the next 5 to 10 minutes. So now, what are the genetic opportunities? Is there a lot of variability in the germplasm? And so I've done quite a bit of work on the grain crops, looking at screening germplasms, collections of the world, and also some wild relatives of the species to see if there is genetic variability. And I'm just going to focus on the pollen germination and the pollen tube growth, because that's the data which I've shown you so far. And there is genetic variability. I showed you some data in the before, but there's larger genetic variability exists. So if you look at the pollen germination, here are some heat tolerant varieties of tomato, where you can see the higher pollen germination even at higher temperatures, and the heat susceptible ones have a lower pollen germination compared to that of the heat tolerant ones. So yes, there is genetic variability available. So this is just a snapshot, but is there a larger genetic variability? There is. So in this case, here is the genotypes. This Dr. Singh screened almost 22 different genotypes and looked at the pollen viability percentage and also the fruit set. So the closed symbols are the fruit set percentage, and then the open symbols are the pollen viability. So you can see some of those lines have very high pollen viability, even at almost 70 to 80%. And then some genotypes have very low pollen viability from 20 to 30%. So in general, if there is a higher pollen viability, there is a more chance of fertilization being successful. So there's a genetic variability in tomatoes. And this is the data on the peppers. And in this case, I'm showing you the temperature response curves. Just like what Dr. Hebar did, all of this data is that they collected the pollen from the different varieties, and then they germinated the pollen in the incubators, which were run at different temperatures. So as you can see, there are three cardinal temperatures: the minimum temperature above which the pollen germination happens, the optimum temperature where you have the maximum pollen germination, and the Tmax, or the ceiling temperature, when there is the pollen germination above which is 0. And in between the base temperature and the minimum temperature to optimum temperature, the pollen germination increases. And then from optimum temperature, the ceiling temperature, or the maximum temperature, the pollen germination percentage decreases. And there is genetic variability, both in terms of optimum. For example, some of those have optimum temperature of 25 degrees Celsius, whereas some have closer to 27, 28. And the same is the case for ceiling temperature. Some varieties had the ceiling temperature of 35 degrees Celsius, whereas some had the 40 degrees Celsius. So there's a large genetic variability for pollen germination percentage. And that is also true for coconut. And in this case, the susceptible genotypes not only have a lower pollen germination and also a lower temperature optimum. This could be around 25, 24, 26, 27 degrees Celsius. And the tolerant genotype has a higher temperature optimum of about 30, 31 degrees Celsius. And of course, the ceiling temperature for the sensitive one was 40, and then the tolerant one was about 45 degrees Celsius. So you can actually screen the genotypes by collecting the pollen and developing these temperature response curves and identifying them, which one of them have higher optimal and higher ceiling temperature to be able to identify the heat tolerant lines. And this is the data on peanuts. This is our own data. So again, the same story. There are genotypes that have huge genetic variability, where you have the variability of the germination percentage anywhere from 70% to less than 35%, and you have different cardinal temperature, and the maximum Tmax ceiling temperature varies anywhere from 46 degrees Celsius to almost 38.6 degrees Celsius. So you can identify those lines by looking at their cardinal temperatures, which are most sensitive. And this is the data from the sorghum. Again, tolerant genotypes have 45 degrees Celsius as the ceiling temperature, and the susceptible genotypes have 40 degrees Celsius as their ceiling temperature. And the major impact of the heat stress has been on damaging the developmental pollen grains, pollen development, and the poor pollen germination. And in this case, the reactive oxygen species story, which I told you. So at a higher temperature, in the susceptible lines, there is much more reactive oxygen species developed, whereas in the tolerant genotypes, the reactive oxygen species is much lower. And that's the main reason for genetic variability of these varieties. And then this is the data on the wheat as well. Huge genetic variability in terms of wheat, and just looking at the pollen germination. And also this improved pollen germination also converts into the increased seed set percentages in those genotypes. So yes, there is opportunities for genetic genetic improvement, and you can use pollen germination as one of the techniques to screen these genotypes. And this is the rice genotype where I'm showing you the data for the spikelet fertility. You have N22, which is highest spikelet fertility at heat stress. And then your L20, L204, L202 are much more susceptible, and there are intermediate types as well. And the main reason for the tolerance for N22 was it has actually produces higher pollen production, and also it has more pollen shed, and in addition to the higher pollen viability as well. So those are the reasons for that. And when you have, and then when you have higher pollen viability and the higher pollen reception, and those genotypes, you have greater seed numbers, and also more filled seeds compared to that of an unfilled seeds. Unfilled seeds are the result of poor pollination, poor pollen germination, poor fertility, therefore no seed set. So that's a genetic variability. Now I'm going to summarize what are the opportunities in terms of managing the heat stress, and I'll talk about agronomic techniques and also some of the breeding methodology which can be used for this, managing the heat stress in horticulture and grain crops. So I don't have time to go into the details, so therefore I'm just providing an overview here. So in terms of stress management, you can use agronomic management. So to do that, you need to identify, look at the last 100 years data for your environment, and look at the temperatures, how they progress through the season, and then when do you typically get the most hottest temperature, and then based on that you can select your planting date and also duration of the variety. The whole idea is to avoid the heat stress happening at those two sensitive stages, at the gametogenesis stage and at the time of flowering. If you are able to avoid the heat stress at those two stages, you can overcome the negative impacts of heat stress. So early planting, you need to make sure that your population is good, plant population is good. If you have opportunities for irrigation, do that, the balanced nutrients, and make sure there's no pests and diseases. All of this irrigation, nutrient management, pests and diseases will keep healthy. If the plant is healthy, it has more energy and more opportunities to either withstand the heat stress or avoid the heat stress. And there are also some chemical options spraying, like hormones, growth regulators, antioxidants, osmolytes, even nanoparticles and microbials, which could be sprayed to the plants before the heat stress occurs. And the whole idea is to improve the antioxidants, minimize the reactive oxygen species, and also regulate the plants so that they can there is more pollen germination, or the gamete viability is not decreased. You can select the plants based on shorter duration, early morning flowering, and this is a trait which has been exploited in rice very much. I don't have time to talk about in detail, but in general, when the day occurs, you have in the morning the temperatures are cooler, and by afternoon or solar noon the temperature goes up. So if you have a genotype or a variety which flowers early in the morning and finishes the fertilization before the daytime high temperatures, that is what is called as early morning flowering. And that is where I identified that in the wild rices, and later on it has been used in the rice. And also that trait is also observed in wheat and also in cowpeas and also in pearl millet. And then in terms of identifying tolerant genotypes, I showed you enough data on the pollen, and there's also opportunities to look at stigma tolerance, and then looking at higher spikelet fertility, rapid grain filling duration, and also looking at stay green types, which can allow you to be more tolerant. So those are some of the stress management techniques. And in terms of breeding methodologies, we have now multiple opportunities to breed for stress tolerance, specifically for heat stress tolerance. You can use the conventional breeding programs, where you screen the genetic resources or the wild relatives or the close relatives to identify the heat tolerant lines. And then you use your normal pedigree breeding and population breeding to develop the heat tolerant varieties. Now we had opportunities for fast breeding or speed breeding, where you can enhance this. You can decrease the time from almost 12 years to 2 to 3 years by controlling photoperiod and temperature. You can have 2 or 3 generations in a year. That's what is speed breeding about. We also have double haploid opportunities for maize and wheat. And then molecular breeding by mapping the genes and then association mapping, marker-assisted selection, and also genome-wide selection are the molecular tools. And although we also have transgenic approaches where you can develop genetic transformation, and then you also have CRISPR-Cas9 technology and the CRISPR-Cas12 now. There's a lot of opportunities and developments examples for grain and horticulture crops using these gene editing tools are available. So I'm at 55-minute marks. I would like to conclude. So hopefully I'm able to convince you that heat stress is a major yield limiting factor for productivity of crops. That includes both horticulture crops and grain crops. And future changes in the climate will further exacerbate this heat stress. And even short episodes of heat stress, anywhere from 2 to 3 days, or even shorter periods of 6 hours, if they happen at the most critical stages of crop development, reproductive stages of development, it can cause significant yield losses by poor floret fertility, leading to lower grain numbers. And food crops, whether it's vegetable, horticulture, or grain crops, are most sensitive to heat stress at 2 stages. One happening at the gametogenesis, few days before anthesis, which impacts the viability of the gametes. And the second one happening at the time of flowering, which impacts the germination, pollen germination, and the fertilization. And then later on, if you have the stress during the fruit growth or the grain growth, it can minimize the grain size. And the decreases in yield and the heat stress is primarily because of the decreased grain numbers or the fruit numbers, but also fruit size can be decreased specifically because of the rapid grain filling duration or the fruit development duration. Crop species and the wild species and the genotype vary in their response to heat stresses and have different tolerance levels. We need to be able to identify those tolerance and use in the breeding program. But however, genetic variability used in the current breeding program is limited. Thus, there is a need to explore systematically screen larger germplasm collections, including the wild species for tolerance, and then use them in our breeding programs. And there is a need to develop high-throughput phenotyping tools and molecular markers so that you are able to screen larger germplasm collection to identify the stress-tolerant genotypes. Because unless you have a high-throughput phenotyping tools, it will be very difficult for you to screen a larger germplasm collection to identify. And also, more importantly, while developing the hybrids or the varieties, it will be very difficult to screen larger germplasm collection. So we need a high-throughput phenotyping tool or a biochemical or a molecular marker for that. And more importantly, as we look into the future, we don't have a very good understanding of the interactions of multiple climatic factors, whether it is temperature interaction with drought stress, or its interaction with vapor pressure deficit, or how does temperature and photoperiod interact, and how does these other abiotic stresses interact with elevated carbon dioxide. Those information of the data is not well known and needs attention. And more importantly, how does the interaction between abiotic and biotic stresses, how does these temperature impacts with weeds, pests, and diseases, and the host plant resistance as well? That is not known, and that needs to be done. And then finally, the impacts of abiotic stresses on the nutritional quality is not well known and limited, specifically in terms of proteins, carbohydrates, fats, micronutrients, antioxidants, amino acids, and vitamins, because food is all about nutrition and health benefits. So how does these abiotic stresses impact these nutrition quality and the health benefits of these fruits and vegetables and grain crops? So those are limited and not well understood, and that needs to be understood well to have a better understanding of the impact of heat stress on food and nutritional security of food and horticultural crops. With that, thank you so much for giving me this opportunity today to speak to you. Sarve Jana Sukhino Bhavantu, Annadata Sukhi Bhava. And then I specifically wanted to thank Honorable Vice Chancellor Dr. Gopal Garu, and also the Director of International Programs, Dr. Dhananjay Rao Garu, for giving me this fantastic opportunity to speak to you today. And I'm available to talk to you and give answers to any of your questions. It could be technical questions or it could be non-technical questions. I know there are a lot of students who are participating in this seminar series. So I'll be glad to answer any questions which you may have. Again, thank you so much for your opportunity to talk to you today. Thank you very much.
Thank you, sir. Thank you very much for your wonderful lecture on impact of heat stress on horticulture and food crops. You have covered almost all areas of greenhouse gas emission, global surface temperatures, heat waves, how the heat stress is affecting the tomato, bell pepper, and coconut, chili pepper, and how the pollen is viability is lost by increasing the temperatures and heat stress, and pollen germination, pollen tube growth, and the interaction of temperature and relative humidity. And I have covered the grain crops also: wheat, rice, sorghum, pearl millet, peanut, and the pollination, fertilization, how it is getting affected with the heat stress and high temperatures, and the mechanism of reproduction failures, effects of heat stress, canopy level, lower grain and harvest index, opportunities for genetic improvement, genetic variability, high temperatures, and creating a lot of variability also, and resistant varieties through breeding, selection, agronomical, chemical, microbial, and tolerant genotypes, molecular breeding, transgenics. So whatnot, everything you have covered. And thank you very much. And excellent presentation. And thank you very much, sir. And now it is open for the discussion. And sir, mostly sir, Varpras sir is from Uyur, Katuru, near Vijayawada. He is our man. So near Vijayawada, Krishna district. So all students and faculty, you can ask our questions and doubts. Any doubts can be clarified from the university officers and all the faculty and all the. I invite our Honorable Vice Chancellor remarks on this occasion.
Yeah, it's a very good topic, and nowadays we all experiencing the rise of temperature, and again there are so many discussions going on about rethinking about crops, and again development of varieties suitable to higher temperatures. So in that context, it is a very good lecture for researchers. Sure. And most of the dry areas, this tomato, it's being grown in larger areas in Andhra Pradesh, especially in Chittoor, Kurnool, Kadapa, and Anantapur. So here, sometimes there will be failure of crops in summer. So probably these are the important factors which disturbs the pollination. So it is a very important lecture, and there is a very need to go for development of heat tolerance varieties with high yield and suitable or have resistance to major diseases and pests which comes in summer. And here our registrar, he worked on tomato for his PhD on heat tolerance. He worked under Dr. Muva Ramagarao Garu, head of the Department of Horticulture. He was the university head also, and he was his student. Yes. And it is very good lecture for all the PG students who are working in vegetable science, and also even in other crops where annual crops are there in medicinal, aromatic, and again in some fruit crops also. So all these mechanisms can be applied. The basics can be applied in development of understanding the things and going for development of varieties in crop improvement. Sir, it is a very good lecture, and really it is nice for youngsters, and I request all the scientists and the PG and PhD students, they can interact with you And again, here once again, I'm stressing who are interested to see if say, everything goes smooth, we can have some kind of a student exchange programs from this year onwards, because recently the semesters have started, first years in both PG and PhD. So we can discuss certain things from now onwards, especially in vegetables and all short duration horticultural crops. Yes. Register Garu, you can continue. And yes.
Thank you, sir. Thank you, sir, for your remarks on the topic given by Dr. Varaprasad Garu. It's really very interesting and very well presented with a very clear, the picture of giving the heat impact on both horticulture crops as well as grain crops, whatever you have presented here. As our Honorable Vice Chancellor told, I worked on tomato for heat tolerance when I was doing my PhD at Rajendra Nagar. Dr. Mova Ramarao Garu and Dr. Vayan Reddy sir, both are my major advisors there. Yes, sir. It was really very interesting to observe your slides. That as you told that this heat stress, it is a common phenomena rather than exception in under Indian conditions. Most of the crops, particularly as our Honorable Vice Chancellor told, in southern region of our Andhra Pradesh, as tomato is a major crop which is being grown on, rain-fed conditions, crop suffers a lot, and the farmers could get very, very meager yields because of the heat stress as well as the moisture stress also. Particularly. As you told that 554 days of heat stress, as a really, it's a long impact on the crops. I think with that, long duration of heat days, I think it will definitely have some this negative impact on the crops. Apart from the pollen grain, the pollination of its one and stigma exertion, and again the fruit formation. These are all the, the affected, the parts and other activity in the croplands. We observed one thing, to mitigate these heat stress, particularly in tomato and other vegetable crops, the hormones, there are some studies, the hormones spraying of some the heat induced some acids, particularly the proline, other contents and other chemicals. Do they have any impact if you spray these hormones during the sensitive stage of the crop? If you provide some life-saving irrigations,
that's right. Yeah.
Do we have any effect on the exogenous application of these hormones to stabilize the sensitive stages so that we can get the normal yields?
Yeah. So what we have done is we worked quite a bit on use of selenium, as it's not a hormone, it's not a growth hormone, but in terms of nutrients, selenium is one of the chemicals. If you are spraying that, what it does is that it increases the antioxidant capacity and it scavenges the reactive oxygen species. So that's one good example. And then the proline example, which you were talking about, is actually an osmolyte. So what it does is that it actually provides the tolerance to the osmotic balance. So under temperature stress or under drought stress, the osmotic balance is maintained. So that is another opportunity. And also there is some information on spraying of cytokinins, the growth hormone, which can protect, because we're all talking about well, specifically when you look at the Gametogenesis, we are talking about meiosis and mitosis. It's all about cell division. So application of cytokines, which is the hormone responsible for cell division, can also provide some opportunities for improving the seed set. Another chemical which is also very important for pollen viability is boron. Boron is also a micronutrient which can significantly improve the pollen viability in terms of boron.
There is a normal, I mean, general practice in our area,
Right.
because cracking will be there in tomatoes, particularly under drought conditions, and farmers will normally use this boron
That's right.
spray. Yes, as you said, that will also help us the pollen, I mean, fertility.
That's right. And then also, I mean, another big one is that when I only talked about the reproductive stages and the gametes and the viability and the seed set. Another one which heat stress, that is rapid senescence. It enhances the senescence of the leaves itself, which are the one which are actually providing the carbohydrates to the fruits. So if anything which you can spray to the leaves to make sure that the leaves are staying greener for a longer duration, can also help with the fruit development in particular. And that's where you have this ethylene inhibitors, because one of the way the senescence happens is the production of ethylene. So under heat stress, the rapid senescence is caused due to increased ethylene production. So if you can spray anti-ethylenes, either ethylene production inhibitors, or even ethylene sensing sprays can enhance the stay green nature of the leaves and can help. And then the last question which you talked about was, if we give some irrigation, can we avoid some of the heat stress? Yes, if you have opportunities to irrigate, you can, because by microclimate of the plant could be impacted, especially the way you are irrigating, whether it is through sprinklers or drip irrigation system. You can make sure that the microclimate is much more, the relative humidity is a little higher. That decreases the vapor pressure deficit, and that also helps, because the coconut example where you clearly saw that the lower the relative humidity, there is a higher damage. But it cannot completely avoid the heat stress, because even if you're irrigating, microclimate will only be there for a few hours. But by the time the midday comes in or the late afternoon, all of your microclimate impacts would have been gone.
Because most of the, if you see your presentation, the night temperatures will play a major role.
Major role, yes.
Major role for the anthesis and also the.
That's right. Yeah.
Set also.
Yeah, so I think because if you look at the reason why nighttime temperature is much more dangerous, much more deleterious than the daytime temperature, is that because if you have the daytime temperature higher, you are only affecting the pollen germination, pollen tube growth, fertilization in terms of reproductive stages. And also you're only impacting the photosynthesis, because photosynthesis only happens when there is light. Whereas when you look at the respiration is the 24-hour deal. It happens in the nighttime, it happens in the daytime. There is maintenance respiration, there is transport respiration, and everything else, right? There's a growth respiration. So the impact of nighttime temperature in particularly is high, because it impacts the respiration rates goes up. And when you have the respiration rates going up, your net available carbon is much more lower. And then the gametogenesis is also 24 hours, whereas pollination and pollen tube growth is only after pollination. Fertilization is completed within few hours or maximum of 4 hours. But once the embryo is formed, it is pretty much stable, or it's much more tolerant of heat stress. So therefore, you see more negative impacts of nighttime temperature is it actually impacts the gametogenesis, whereas the daytime temperature is only impacting half of the time, or only the pollination and the pollen tube growth. And that's why the early morning flowering is a good trait. Those flowers in the nighttime or those flowers in the early morning to avoid the heat stress, that's a pretty good trait to have. It's an escape mechanism. It's not a true tolerance. Escape. Because if you look at cowpea and even pearl millet, the fertilization and everything is happening in the nighttime, not in the daytime. So if you can identify tomatoes or peppers or those lines, which is where the pollination and the fertilization and the pollen tube growth is happening in the nighttime or early morning, then you have an escape strategy. And fortunately, I think the early morning flowering is a trait which is controlled by fewer genes rather than more number of genes. So there is an opportunity for genetic improvement for that.
Thank you. Thank you, sir, for your nice.
Thank you, sir, Register Garu. And any others, university officers or faculty, students, almost 1.5 hour time is over. Anybody?
Sir, myself, Dr. Seshkiran, sir. I am from Horticulture Research Station, Venkatraman. Sir, you mentioned about variation in the varieties for the heat stress. Is it due to genetic makeup or is it due to the endophytic bacteria in the particular varieties that is giving the heat stress tolerance, sir? Or it can be both?
Right now, most of our information is about genetic trait because we have not really looked into the microbials. It could be endophytic bacteria or the microbials. Therefore, the microbiome of the heat-tolerant varieties, both from the roots perspective and also from the plant perspective, both ectophytes and the endophytes, that information is not available. It's not really known. So far, whatever I'm showing you seems to be more of a biochemical mechanism. So yeah, the data which I have shown you is primarily genetic differences, but we don't know if that genetic differences is in response to endophytic bacteria or either that's another area of research which is needed.
Sir, one more question, sir. As you told about some amino acids and peptides.
Wait
Yes,
for
sir.
1 minute. Sir, we are going to attend the Diamond Jubilee celebrations of Acharya Engineering Agricultural University. So we have at 3 o'clock you have to join meeting there. Yesterday it's inauguration and everything is over. So today we have some kind of panel discussions and all. And myself, registrar, and some of the university officers leaving to the meeting. So kindly continue. Thank you, sir. We look forward for your collaboration and strengthening the MOU and all with these between these two universities. Thank you, sir. Thank you.
Thank you for your time. I appreciate it. Yeah, thank you. Thank you.
Thank you, sir. Honorable Vice Chancellor, sir. And
thank you.
Can continue.
Dr. Dharanja Rao.
Sir.
With your permission, I will also take leave.
Yes,
because I had to accompany our Honorable Vice Chancellor to that Diamond Jubilee celebrations.
Yeah, please convey my regards to the University Leadership Sharada Garu and Srinivas, because I joined Bapatla and I was there for a year before I moved to Rajahmundry. So most of my colleagues are now university officers in
Sure, sir, I will convey.
Thank you,
sir. Thank you, Dr. P. Srinivas Garu, Registrar. So we can continue for a few more minutes, sir. Please, anybody want to ask?
Sir, in continuation of my previous question, sir, you told about the amino acids and peptides. We see a lot of these products like from Syngenta international brands like Isabian or Quantis. So they are full of amino acids, peptides, and also some seaweed extract. So are they going to give any mitigating measures during this heat stress time? If we can rescue the crop, particularly during that heat period, and say, take an example, you have mentioned about the tomato, where we have very significant acreage under that particular. So majority of the farmers, they take up during April or so. So if you can alter that, protect the crop till flowering stage, like say by planting in May or April ending. So and because as per your presentation, it is going to affect the only the reproductive stage. So if you can rescue the crop till that vegetative stage, it will be good. And moreover, being all these have multiple pickings and all the impact of heat stress will be present throughout the crop rather than during a particular season, particular period, like brinjal or tomato or chilies.
That's right. Yeah, I mean, that's a great question. I think if you can avoid the heat stress during these key stages of crop growth and development, especially at the gametogenesis and the pollination or anthesis, then you have more opportunities for seed set. That's one way of doing it. Another one is that if you can also, because, as you mentioned, these are indeterminate crops, right? So you continue to get new flush of flowers. So if you have tomatoes or other crops which can flower for a longer duration, so that you have an opportunity for the next set of flowers to which can avoid the heat stress, good farm seed set and farm yields. So anything you can do to mitigate or avoid or spray to tolerate the heat stress during those sensitive stages would be of high value.
The majority of your slides, you mentioned that heat stress is giving at 30 to 35. If the damage is occurring at 32, we can the damage that is expected in this particular tropical conditions, where in summer the temperatures are rising beyond 45, we can expect more damage. Where in your experiment it is only 35 degrees. It is a heat stress.
Yeah. I mean, these are very sensitive. Yeah.
Thank you so much for
that. Nowadays, if you, I'm looking at I was talking to my dad yesterday. So he was saying almost the temperatures are reaching 30, 38, 40, 42 degrees Celsius. Daytime high temperatures. Yeah.
Sir, it was a nice presentation, sir. Majority of you focused on the implications of that. If you would like to, if you have any work carried out on the management aspects, one more lecture on that will be great.
Yeah, so I only focus on reproductive stages and gametogenesis, but we are doing quite a bit of work on management strategies. We are using crop simulation models, primarily looking at the last 100 years worth of data in terms of when does the heat stress happens, as the occurrence of the heat stress events moving earlier in the season or later in the season, and then also because most of the crops you can select a maturity group: early maturing, medium maturing, late maturing. So the idea is to match the genotype with the environment and then develop the management strategy. So we're looking at G/E/M interactions to avoid the heat stress at sensitive stages. And then I only focused on the male reproductive parts, mostly on the pollen germination, but also usually the question I get is it the pollen which is more sensitive or is the ovule which is more sensitive? So if I've looked at majority of the crops in general, it is pollen which is much more sensitive to heat stress compared to that of an ovule, because pollen, once it is dehisced, it is independent. It's not connected to the mother plant, whereas the ovule is always connected to the mother plant. So it can actually, because it is connected, it can regulate its temperature or moisture and other stuff. So pollen is in general much more sensitive to heat stress.
So one more lecture on your management will be very great, sir, because in we working in this horticulture universities, majority it will be farmer centric. So any recommendations?
Next time I'll come, when I come, I'll talk a little bit more about management.
So most of the universities do applied research.
That's right. And I think I wanted to tell you this, but all of these things which I've showed you is that it doesn't require a whole lot of facilities to conduct this research. All you need is an incubator where you can have these temperatures grown, and then you tissue culture. So just agar medium, you collect the pollen, put it in the incubator, get the temperature response curve. So it's not very expensive equipment. It's to do the pollen, but of course the antioxidants, the reactive oxygen species and enzymes and all will take a little bit more. Lipidomics and metabolomics will take a little bit more funding, but doing simple pollen germination and developing temperature response curves is not an expensive process. It's more of an art. If you can get the germination media right, you'll be able to do that. So I see that there's a question from...
Thank you, Seshkaran. And anybody from the student community or faculty?
There is a question in the...
Sir, myself, Dr. Nalini, from College of Horticulture, Venkatramanagodam. So you have mentioned about the reactive oxygen species increase may cause reduction in the antioxidants and cause stress to the plant. But as pest management concept, we take the host plant resistance concept. As you are already mentioned in the conclusions, that interaction between the abiotic stress and the biotic stress is not done yet, the research on it. But most of the time, we considered for the tolerant crop or resistant crop, reactive oxygen species will induce this transduction in the plant against the pathogen and the pest. So increase in the reactive oxygen species is one mechanism where the plant is ready for the defense if it's defense against the pest and
That's right.
pathogen.
That's right. So it's a great question. So in general, reactive oxygen species, there's under normal conditions or in optimal conditions, there is a balance between the reactive oxygen species and the antioxidants. Reactive oxygen species are needed in the plant. You cannot completely stop because those are the signaling molecules. And just as if you were mentioning about the insects and the disease, they serve as a signaling molecule so that once the insect or the disease falls in, the reactive oxygen species goes up, and then it gives a signal, and then there could be the plant will respond to that by releasing some other chemicals to protect their membranes and other stuff. So in general, in a normal condition, there's a balance between ROS and reactive oxygen species and
Yes.
antioxidants. But under heat stress, what happens is that ROS goes up and then the antioxidant comes down. So there's much more ROS at a higher concentration is bad for this. And I was specifically talking about the reproductive tissues, whereas what you are talking about when it is about insect and the diseases is that the concentrations are not so high. They're much lower, and that triggers the protective mechanism saying that yes, there is a pest coming in, or the intensity of that host plant resistance needs to be increased. So yes, it is important. ROS is certainly critical, but they should not cross a certain limit where it could be damaging the membranes.
Yes. Thank you, sir.
Let me take a question from the chat box. There is a question in the chat from Dr. Kanchana.
Student ID is marked with the elder man.
Yeah, she's asking that what are the opportunities for postdoctoral studies at Kansas State University? So I know there is a lot of students who are present in the audience. So let me talk a little bit about the opportunities for graduate students, and then I'll come to what are the opportunities for the postdoctoral opportunities. So we're always looking for good graduate students. For example, last 5 years, I bought more than 175 undergraduates from India, specifically from Angra, Acharya Engineering Agriculture University, 62 students from there, and almost 30 students from Sher Kashmir and other places from India, mostly undergraduate students, for a period of 60 to 90 days. And this was a NIHAP program. Dhananjay Ragharu probably knows about it. This was a ICAR and World Bank funding. So many of the universities got funding for that. That was the first round. It was total of $63 million from ICAR and $60 million from World Bank. So it was a lot of effort. And there could be a new phase for that coming up in the future. So I would certainly encourage YSR Horticulture University to compete for that. And then through that, what I bought, I only focused on undergraduates. So we bought undergraduate students for, as I said, 60 to 90 days to provide them training on whatever their topic of interest was. And then when they come here, they work with the different faculty members in the College of Agriculture or Horticulture. And then that creates a connection and the link between the professors and the students. And many of those students return back to do their master's program. We recruited almost 7 to 8 students from that group, whoever was interested to continue their master or to do their master's program at K-State. We were able to provide them full scholarship and give them the admissions. So there is opportunity for that. If your undergraduate students would like to come to K-State for a 60 to 90 days, we can see your performance, we can see your aptitude for research, and then there could be opportunities for higher for masters. And if you're already having a master's program, then you can come over for the PhD program. And the two important things which you need to apply for either master's or PhD is, number one is a TOEFL, meaning that your English ability to speak and write and comprehend English. And there are two types of tests, TOEFL or IELTS. US universities prefer TOEFL, whereas IELTS is preferred by the UK or the European system. You can take either one, but if you're starting, I'll certainly request you to take a TOEFL rather than IELTS. But if you have taken IELTS, then those scores could be converted into TOEFL. There is a formula they use to convert that. So that's a must. You should get a minimum score to be able to get the admission. The second one is called GRE. And GRE exams are important to make sure that scholarship is given to those students who have high GRE scores. So those two are the minimum basic requirements. And then also you need to write a statement of purpose, which is research statement. Why do you want to select Kansas State University? Why do you want to do horticulture? And why do you want to do physiologies? I'm just giving you as an example. You can pick your own subject area and then pick that topic. So it's a one-pager typically saying why Kansas State, why that college, why that department, and why that specific area of interest. Those, and then that's important. And the final one is your curriculum array or CV, targeted CV, which all your educational background, any awards, any leadership activities, any poster presentation, oral presentation or publications, short-term training, long-term training, all of those. So you need all these four: GRE, TOEFL, statement of purpose or research, and your CV to be able to apply that. And then we usually take admissions in 2 times. One is called fall semester, which starts in August, and then the August of every year. And then the deadline for those applications is the previous December. So if you want to come this August, the deadline for our application would have been the last December. And we typically make the decision on scholarships and everything right about between March and April. So that's one admission. Second admission is spring admission. The admissions start in January, and the application deadline is sometime in this spring, May, June. And the admission will be given July, August, and then you'll start your program in spring. So those are the 2 major types, and most of the information is available on the websites. So that's for the students. And nowadays we only, if you are coming for a degree seeking, we use the F1 visas, which is a student visa. But if you are coming for a shorter duration, just like the students which I brought from India on a 60 to 90 days, we usually bring them on J1 visas, which are called visiting scholars. So that's a short-term visiting scholar. And once you have that, because you're on a short term, you can return back to your home country, finish your degree, and then come back on an F1 visa. So that's for the undergraduates and the graduate students. And this specific question was focused on postdoctoral opportunity. So postdoctoral, there are certainly postdoctoral opportunity. The primary, you don't need a GRE for postdoc. You just need a TOEFL. TOEFL is must, whether you're applying for a graduate school or postdoc, TOEFL is a must. And if you don't have a TOEFL, then you'll have to take an interview. So there'll be our university has an English program. They'll set up an interview with you. And then once you pass that interview, then they'll allow you to apply. But for postdoc, the most important criteria, the way we select postdocs is your publication record. Were you able to publish from your master's research? Were you able to publish from your PhD research? And if you're employed after PhD, how have you published your data and the quality of those journals, that is impact factor of those journals. So those are very important.
Thank you.
And postdoctoral research scholars are typically bought on J1 visas, which is a visiting scholar visa. If you're less than 6 months postdoc, then we'll do a regular J1. And if you want to do a longer term postdoc, for example, more than 6 months or in a year, we can bring you as a visiting scholar with a longer duration. So there are 2 types of J1s. One is shorter version, and then another one is a longer version. So if you're using the longer version, you can stay in the country on a J1 for between 1 to 5 years. Yeah. So publications are the key for postdoctoral.
Thank you, sir. Thank you very much. And anybody almost 2 hours is over. And student community, any students, it is
I have a
also,
question.
as said by the sir, that the director of industrial and international program already conducted 23 Global Connect series. We have different scientists from different countries. The students can contact me for their phone numbers and after consulting them and by phone, you can go abroad for any studies or any research work, any for any employment. Our university is establishing linkages with the international scientists and industry also. Some students who are going to complete BSc Agriculture, they can observe in private industries also. That we have conducted chambers of Indian industry and AP chambers of Andhra Pradesh. So you can go to directly in industries. We have phone numbers. So employment opportunities and for internship also we can have with industries in Andhra Pradesh and Telangana. So no problem. We are establishing linkages between the university, industry and international community. So you can avail this facility from Director of Industrial and International Programs. So if nobody is there, we can conclude. And rapporteurs, Dr. Nalini, Assistant Professor Entomology, College of Horticulture, V.R. Gudem, and Dr. S.B. Ramya Lakshmi, Assistant Professor Agronomy, Economics, College of Horticulture, Anantarajpeta, Archana, TO2 DIP. So please kindly note down all the minutes and submit. And finally, anybody is there? Want to talk or we can close?
Sir, I have a question, sir.
Okay.
Sir, good morning, sir. Thank you for pressing the major issue, which is regard to the climatic change. Sir, I have a question. You have covered all the crops which are mainly reproductive biology concerned, like the crops which are having economical yield of a biomass. Like we have to target on genetic improvement or the crop improvement. Like what will be the traits we are supposed to, I'll say, target? Like you said, early flowering in terms of tomato. Like for like crops like turmeric, ginger, and we have ashwagandha or sarpagandha. Those are the crops which are mainly focused for the biomass yield. In this crop, heat stress is a serious issue, and these are mainly linked with the chemical compositions, and they have serious phenotype into environment interaction. This is the first question, and I have other question. You have said, yeah. Another question I have said, like you said, pollen is the most sensitive issue in tomato. Like we usually study about receptivity is the serious question with regard to the stigma. Yes, sir. And then third topic I have, like I was looking for postdocs in abroad. I have tried with Alabama, Auburn, Purdue, and all the universities. Should I target the work which I worked on PhD, or should I deviate my subject to other journals? Like that is the major issue. I have tried and failed several instances with Texas A&M also, with other countries also. Thank you, sir.
Sure. Great questions. So let me answer your technical questions first, and then I'll come back to your application on the postdoctoral opportunities. So the first question was, yes, most of the data which I presented you or I worked on was on the grain or the fruit crops where reproductive tissue is a primary impacted by the environmental stresses. But you're absolutely right that there are other crops where the economic yield is not a fruit or a result of a fertilization, especially if it is a tuber crops or if it is a leafy vegetables or other like, for example, biomass crops. So in those crops, the biggest impact of heat stress is going to be on photosynthesis, availability of the photosynthesis. One of the meaning availability of the photosynthesis itself, because as I showed you, one of the methodology or one of the ways the heat stress impacts is the decrease in photosynthesis, decrease in green leaf area duration, and also partitioning of that carbohydrates to the economic part. So if you want to screen genotypes for those crops, the thing which you need to look at is the stay green, meaning that is there a genetic variability where the impact on the chlorophyll content on the chlorophyll fluorescence of the leaves is green leaf area duration is longer, or they are senescing slowly. And one way to measure is using a SPAD meter, quick measure, or NDVI. If you want a larger germplasm collections, you can fly a drone and measure the NDVI, which will actually give you the greenness of the leaves, which is an indicator of the stay green trait. Or you can use the chlorophyll fluorescence, which will give you the damage to the membranes. And then that's one trait, stay green or longer green leaf area duration. And the second trait is that canopy temperature depression. So if there is a genotype which is avoiding the heat stress by keeping the canopy cooler, that's called canopy temperature depression, meaning that when the stomata are open, and if you have irrigation, the evapotranspiration will happen and the leaf temperature will come down. So if the leaf temperature comes down, then the senescence will be slower, or the growing degree accumulation will be slower. So that would be another trait which is called as escape of canopy temperature depression. And you can evaluate that trait by thermal imaging camera or imaging just a regular thermal camera. Would be useful. So those lines which are keeping the canopy cooler are the one which are avoiding the heat stress. So that would be the second trait which I will go after. And the third one is going to be the partitioning, meaning that how much of this, meaning that, for example, if you're looking at the root or in the other biomass, so you actually do the root shoot ratio, or you look at the economic part and the total biomass, and then making sure that carbon and nitrogen from the leaves and the stems is actually going to the economic part. So those will be the three traits I will use: stay green, canopy temperature depression, and greater partitioning to the economic parts, whether it is stem or roots or leaf itself, biomass itself. So those were the first technical question. The second technical question was, you talked about pollen versus stigma. And most of the work, as I said, I think I already responded to that question, saying that in general, for most of the crops, pollen is much more sensitive compared to that of a stigma. And the reason being that once the pollen is dehisced, it's an independent, it's not connected to the mother plant, whereas stigma is always connected to the mother plant. So there is opportunity for it to be a little bit more tolerant for heat stress. So that was the second technical question. I know you had another third technical question, which was probably something related to spraying. I'm not sure, but I'll go ahead and ask.
No, sir. Like, what are the traits we are supposed to target? Like biochemical markers. Do you have any other works on these aspects?
Yeah, so the traits, as I said, stay green, canopy temperature depression, greater partitioning, higher seed set, higher pollen viability, shorter grain filling duration, higher grain filling rate. When I say grain, it is also translated into fruit, longer fruit, and then also more flowering, more flushes of flowering. So if your first flush of flowers is impacted by heat stress, can it produce a second set of flowers, which could happen in a cooler time frame, and they could farm the yield. And also root traits as well. If you are much more profuse, rapid root growth, so that you'll have better moisture absorption and also better nutrient absorption. So those would be a lot of traits. And I talked about physiological traits, right? You can also look into the biochemical traits or molecular markers or biochemical markers, especially looking at the oxylipids, and also looking at the reactive oxygen species, or also antioxidants, or also osmolytes. So there's a lot of these biochemical traits which could be utilized for screening the stress tolerance. So now coming to your question related to the postdoctoral opportunity, and you said that you applied at Texas A&M, you applied at Auburn, you applied at Purdue and were not successful. And then what is the topic which you should be pursuing for postdoc? So let me first respond to the first question. As I said, the methodology for screening the candidates for postdoctoral is purely your publication record and the number of publications and the quality of your publications. So if you have not been successful in publishing your master's research, or if you have not been successful in publishing your PhD research in a good quality journal, then it will be difficult for you to get a postdoctoral opportunity. But you can,
do you look for the Sir, I have small question. So do you look for that quantity or do you look for the quality, sir?
It's a combination. It's a combination. Preferably, you want it to be an international journal. Numbers doesn't matter if you have one, because nobody expects it. Well, when you're looking for a postdoc, I'm assuming that you don't have you're a fresh graduate, or you just started your early career as a faculty member, right? So those are the people who typically look for postdoctoral opportunities. So we don't expect a whole lot of, in terms of number, we don't expect a whole too many publications. So typically, most of our students, if you are a master's students, you get one good quality publication. Or if you're too good, then you can get two good quality publications. If you're a PhD students, you're expected to have two papers, and good students will have 3 papers. So one for master's minimum, and then 2 for PhD. That's good enough. But if you are, let's say, for example, if you're publishing in Andhra Pradesh Agriculture Journal or your university journal, it's not that valuable. Or if you're only publishing in a national journal like some Indian journal, then it won't be good. So I think the quality is important, meaning that you should publish in an international journal. It could come from India. I'm not saying that it can't be an Indian journal. It could come from India, but it should not be only for India focus, because a lot of Indian journals are also accepting papers and are publishing from international audience as well. So as long as the journal is an international and is published by a reputed publisher, that should be okay. So numbers are not that critical. 1 or 3 publications for postdoctoral application is good enough. The quality is much important. And another question was what subject area, right? So I tell to my students that you have 3 opportunities to decide where you want to go or what you want to do. First one is once you're doing your master's research. Let's say, for example, if picked crop physiology or horticulture physiology for your master's program. When you are going from master's to, and you realize that, hey, this is not what I want to do. I want to be doing something different. So your second opportunity to change your subject matter is when you're applying for a PhD. So you could have done master's in physiology. It doesn't mean that you have to continue doing your PhD in physiology. You can do your PhD in agronomy. You can do your PhD in grain science. You can do your PhD in plant breeding and genetics or biochemistry. So transdisciplinary nature, those days are gone when you start your master's in physiology and you end up as a faculty member in physiology. Those were 20 years ago, 15 years ago. Now it's all transdisciplinary, so you can change your field. That's not at all looked at as a negative. And the third opportunity which you have is once you have a PhD and you realize that, hey, I don't think I'm going to continue in this area, your postdoc is your next opportunity to change your subject matter. And that is the most critical. You don't want to do 2 or 3 postdocs. And so that when you apply for a postdoc, choose an area where you want it to spend your rest of your career, 20, 30 years in that specialization. So, and then doing a postdoc on your PhD work is not a good suggestion, because in a postdoc, you want to learn something new, something different, what you have not learned in your master's program or in your PhD program. So postdoc is an opportunity to develop new skills, understand new science, so that you are up to date in your technological advances which are happening in your subject matter. So I would not apply for a postdoc in an area of your own specialization. If you're doing that, you're not benefiting yourself, you're benefiting the faculty with you are working, because he would want you to, because you are specialized in that area. And for example, if you want to come to my lab and do reproductive biology, and if your PhD is in reproductive biology, I'll take you in a split second, because it's benefit to me, because you have those skills and I'm working on that. So it will be a benefit. It will be much more benefit to me. But if you're already trained in reproductive biology and my work is on reproductive biology, why would you come to me? It's not going to be any beneficial to you. So you would want to go to a professor from where you can learn rather than you can give. So that trade-off is there. So I don't know what you applied in Auburn, Texas, or in Purdue. So I would certainly encourage you to make a case saying that, hey, I did my master's and PhD here, and I'm a new faculty member, and this is where I want to go. But these are the skills which I don't have it, and therefore I know that your work, your lab has this expertise, and I would like to spend a little bit of more time to learn from you so that I can be successful in my professional career as a faculty member. And I would like to build a longer term relationship with you or collaboration with you so that even when I come back after the postdoctoral opportunity, I would like to continue the collaboration. So that's an approach you would certainly want to take.
Thank you, sir. Thank you so much.
Thank you, sir. Thank you very much. Almost more than 2 hours over. I think yours
Yeah,
is
it's also
1 o'clock.
almost 2 a.m. 2 a.m. for me. So
Anyhow, you are coming in June. So
yeah.
definitely we will have one more lecture, and through Zoom also we can contact our students and faculty in June. We are eagerly waiting for your journey
Yeah,
here.
I'll confirm. I've not yet fixed my dates because all the things which is happening with the federal government in US right now, we are juggling a lot of balls here. So we have to we will. Yeah, once things settle down, my plan is to come sometime in the first week of June and then be there for the entire month.
Okay, sir. Thank you very much for excellent presentation, nice presentation, and answered question answer. Almost, I think our students and faculty doubts are cleared. And thank you very much, sir. Once again, from Dr. Vaisar Horticulture University, you have taken a lot of patience and more than 2 hours time continuous talking with our faculty and students. And I thank you once again. And I thank all our university officers, especially our Honorable Vice Chancellor, who has taken a lot of pains for conducting this program. And I thank our Honorable Vice Chancellor and our Registrar, Dr. B. Srinivasulu, and all our university officers and all our faculty, associate deans, heads of research stations, heads of KVKs, and the principals of polytechnic colleges, and all our students, especially who are actively participated in this 23rd Global Connect series. Thank you once again all, and I also thank our rapporteurs, Nalini and Ramya Lakshmi, Archana. So I thank you once again, sir. Thank you all for participating this program. Thank you once again. Thank you very much.
Thank you. Have a good day. Take care.
So now the program is closed.